Hello and welcome to the first lecture of the Module 3. Today we are going to discuss the role of plants in the environment. Life, as we know it on our green planet, would not be possible without plants, as they are considered to be the critical resource for life sustainability because of the many ways they support life on Earth. They release oxygen into the atmosphere, absorb carbon dioxide, provide habitat. A single tree may provide food and shelter to many species of insects, worms, small mammals, birds, and reptiles, as well as food for wildlife and humans, and, of course, regulate the water cycle, accounting for 10% of the global transpiration. By one definition, plants form the clade Viridiplanta, Latin name for green plants, a group that includes the flowering plants, conifers, and other gymnosperms ferns, as they are alleys, hornworts, liverworts, mosses, and the green algae, but excludes the red and brown algae. Green plants obtain most of their energy from sunlight via photosynthesis by primary chloroplasts that are derived from endosymbiosis by cyanobacteria. Their chloroplasts contain chlorophylls A and B, which gives them their green color. Some plants are parasitic or mycotrophic and have lost the ability to produce normal amounts of chlorophyll or to photosynthesize. Plants are characterized by sexual reproduction and alternation of generation. Also, asexual reproduction is quite common. There are about 320,000 species of plants, of which the great majority, some 260,000 to 290,000, produce seeds. Plants that produce grain, fruit, and vegetables also form basic human foods and have been domesticated for millennia. Plants have many cultural and other uses as ornaments, building materials, writing material, and they have been the source of medicines and psychoactive drugs. Talking more about the role of plants in the environment, it will be good to talk about the other benefits that we get from these plants. These plants help in preventing the soil erosion. They hold the soil in the best manner. They also lessen the chances of soil infertility. Plants continuously add the minerals to the soil. They break the larger components of the soil into smaller ones. These are not only used by the plant itself, but the other organisms living in the soil also rely on it. Plants are also a source of industrial products. Wooden furniture, doors, windows, and different household products are from wood of huge trees. Plants are a great source of medicine, even for life-threatening diseases. More than 100,000 natural compounds come from plants, and most of these have yet to be explored. Also, plant medicine is safer due to the lower chances of side effects and better compatibility to humans. Medicines derived from plants include vincristine, digitalis, colchicine, reserpine, quinine, morphine, taxol, and aspirin. Some plants are also grown for the sake of biofuels. Plant products are used as coal and other fuel products. The plant's fuel is less toxic as it does not emit harmful gases and are also less expensive. Even the plant waste is used to generate electricity. Plants are also the source of insecticides and pesticides. Artificial pesticides are harmful to the environment. They may even enter food, causing health issues to humans and animals. Using these naturally derived pesticides are safe. They degrade with time and even do not harm the soil. As we could see today, we obviously can't live without plants, but sometimes they cause us problems. Many plants are weeds and parasites, which means they are the plants which grow where people don't want them. They take up the space and use resources, hindering the growth of more desirable plants. People often introduce plants to new habitats where they lack natural predators and parasites. The introduced plants may spread rapidly and drive out native plants. Many plants produce pollen, which can cause allergies. Plants may also produce toxins that harm human health. Please feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and looking forward to seeing you soon. AJ. It is remarkable how 20 years have changed the concept of plant signaling, from a situation in which even the word signal and transduction received no mention, to special issues devoted to the critical topic. Signal transduction is used by plants to coordinate their development and to sense and respond to fluctuations in their surroundings. A substantive number of genes will be involved with the signaling necessary to specify cell position and fate in developing leaves or roots. A plethora of signaling molecules for cell development must be present with their transduction machinery, because cells continually change position during growth and must continually update their spatial and temporal conditions. Although sugars and auxins may be simple molecules, they have long been seen as signals in vascular tissue differentiation. Furthermore, other complex signal transduction circuitry must be present to ensure the stability of morphogenesis, the overall form of leaves, roots, and flowers. In chaotic environmental conditions, although some plant characteristics are resistant to environmental variation, many others respond plastically, adding an extra layer of morphogenetic and biochemical complexity that is largely unique to plants. Cytokines, opsicins, ethylene, and gibberellins, as well as more recently discovered barcinosteroids, jasmonate, and peptide hormones, are agents that help to strengthen plasticity. As they are resistant to environmental variation, many others respond plastically, adding an extra layer of morphogenetic and biochemical complexity that is largely unique to plants. respond plastically, adding an extra layer of morphogenetic and biochemical complexity that is largely unique to plants. Cytokines abscissins, ethylene, and gibberellin. From a situation in which even the word signal and transduction received no mention, to special issues devoted to the critical topic. Signal transduction is used by plants to coordinate their development and to sense and respond to fluctuations in their surroundings. A substantive number of genes will be involved with the signaling necessary to specify cell position and fate in developing leaves or roots. A plethora of signaling molecules for cell development must be present with their transduction machinery because cells continually change position during growth and must continually update their spatial and temporal conditions. Although sugars and auxins may be simple molecules, they have long been seen as signals in vascular tissue differentiation. Furthermore, other complex signal transduction circuitry must be present to ensure the stability of morphogenesis, the overall form of leaves, roots, and flowers. In chaotic environmental conditions, although some plant characteristics are resistant to environmental variation, many others respond plastically, adding an extra layer of morphogenetic and biochemical complexity that is largely unique to plants. Cytokines, abscissins, ethylene, and gibberellins, as well as more recently discovered barcinosteroids, jasmonate, and peptide hormones, are agents that help to strengthen plasticity. As the environmental context changes, so will the strength of connections vary between the proteins and other molecules that make up the network. A strong spatial dimension underlies signal transduction. For example, plants must be able to detect gradients in signals, such as light, and resources, such as nitrate and water. Plant development itself also is decidedly polar. The spatial dimension is satisfied in many ways. Cells place receptors, channels, G proteins, and kinases in specific membranes. Some signaling protein complexes are permanent, such as the relatively stable and perhaps hardwired constitutive photomorphogenesis signalosome COP9. Other signaling protein complexes are likely to be ephemeral and formed immediately as a result of signaling. There are at least 600 receptor kinases in Arabidopsis, and most of them are membrane-bound. Incompatibility and disease defense signal transduction use receptor kinases. After ligand binding and autophosphorylation, such kinases may act as nucleation sites for the construction of ephemeral signaling complexes that contain many proteins. Many other GTPases, 14-3 proteins, kinases, and phosphatases may cluster around these nucleation sites, forming kinase cascades, and the regulation of specific transcription factors, for instance, biphosphorylation, will complete the pathway. Please Feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and looking forward to seeing you soon, AJ. Biotechnology is the use or manipulation of an organism or the components of an organism. Plant biotechnology is a powerful tool for the development of new plant traits and varieties. Such new varieties must be produced on a large scale to achieve commercial success and to satisfy the demand from growers. Traditionally, new varieties were achieved by the seed propagation method. Currently, micropropagation produced plantlets offer a practical alternative for many plant species. Micropropagation is labor-intensive, although the time needed to commercialize new varieties and permit the production of disease-free plants is reduced. Recent developments in genetic engineering and molecular biology techniques allow the production of improved and new agricultural products in several countries worldwide. This is only possible with the evolution of tissue culture techniques, which provide the platform for the introduction of genetic information into plant cells. Current plant biotechnology has developed as a new age of science and technology, where production of secondary metabolites, valuable plant genetic improvements, germplasm conservation, and production of large number of disease-free and new varieties are preferred. Production of artificial seeds, biopharmaceuticals, plant-made pharmaceuticals, recombinant or other therapeutic proteins, transgenic plants, and plant-made vaccines or antibodies, plantibodies, is part of the current research work in plant tissue culture science. Most of the plant tissue culture industries are exploiting the fundamental property of regeneration of plant cells for rapid production of elite varieties with superior genotypes on a large scale in a comparatively short time. Initial problem: search for isolated cell model, validation of plant cell totipotency, identification of the role of growth substances, development of many techniques, transgenesis. The current plant tissue culture industry is estimated to be worth around $150 billion, 50 to 60% of the agriculture industry. Annual demand of tissue culture-raised products is about 10% of the total estimated value, is $15 billion, with a growth rate of 15%. Most of the plant tissue culture-based industries are limiting the time-consuming labor or manual processes fully by transforming into more automated and robust technologies. The introduction of such robust and automated technologies has led to rapid multiplication of plantlets at a larger level. Please feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and looking forward to seeing you. Plant breeding is the science of changing the traits of plants in order to produce desired characteristics. It has been used to improve the quality of nutrition in products for humans and animals. Plant breeding can be accomplished through many different techniques, ranging from simply selecting plants with desirable characteristics for propagation to more complex molecular techniques. Genes in a plant would determine what type of qualitative or quantitative traits it will have. Plant breeders strive to create a specific outcome of plants and potentially new plant varieties. Plant breeding has been practiced for thousands of years, since near the beginning of human civilization. It is practiced worldwide by individuals such as gardeners and farmers, and by professional plant breeders employed by organizations such as government institutions, universities, crop-specific industry associations, or research centers. International development agencies believe that breeding new crops is important for ensuring food security by developing new varieties with improved quality, such as higher yield, increased nutrition, improved flavor, disease resistance, regional adaptation to different environments and growing conditions, as well as longer storage period for the harvested crop. One major technique of plant breeding is selection, the process of selectively propagating plants with desirable characteristics and eliminating or culling those with less desirable characteristics. Another technique is the deliberate crossing of closely or distantly related individuals to introduce traits or genes from one variety or line into a new genetic background. During infection, pathogens secrete an arsenal of molecules collectively called effectors, key elements of pathogenesis, which modulate innate immunity of the plant and facilitate infection. Among the R genes displaying complex interaction with AVR genes, some of the most promising are those conferring broad-spectrum resistance R gene, since they guard key components of plant immunity and as such target essential effectors, are proteins. Another promising strategy to manage durable resistance would be to target antagonistic interactions between AVR genes and to combine the corresponding R genes in the same cultivars through pyramiding or to sequentially use the R genes in rotation. Although antagonistic interactions between AVR genes have only been reported twice in plant fungi pathosystems, they are probably more widely distributed than suspected. Indeed, in cereal powdery mildews, it has been suggested that pairs of AVR genes and suppressors of AVR gene recognition could form the basis of specificity. This research is facilitated by revealing the molecular mechanisms of plant resistance in plant breeding. For example, a mildew-resistant pea may be crossed with a high-yielding but susceptible pea. The goal of the cross being to introduce mildew resistance without losing the high-yield characteristics. Progeny from the cross would then be crossed with the high-yielding parent to ensure that the progeny was most like the high-yielding parent, backcrossing. The progeny from that cross would then be tested for yield and mildew resistance, and high-yielding resistant plants would be further developed. The classical plant breeder may also make use of a number of in vitro techniques such as protoplast fusion, embryo rescue, or mutagenesis to generate diversity and produce hybrid plants that would not exist in nature. Please feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and looking forward to seeing you soon. AJ. Pathogenic bacteria display various levels of host specificity or tropism. While many bacteria can infect a wide range of hosts, certain bacteria have strict host selectivity for humans as obligate human pathogens, determined by multiple molecular interactions between the pathogens and their host. Understanding the genetic and molecular basis of host specificity in pathogenic bacteria is important for understanding pathogenic mechanisms, developing better animal models, and designing new strategies and therapeutics for the control of microbial diseases. This is exemplified by the recent development of meningococcal vaccines based on the VH-binding protein of Neisseria meningitidis. In this case, selectively inactivating the FH-binding activity of the FH-binding protein led to enhanced immunoprotective efficacy of the vaccine. Because of the CBPA, PCPA, and IgA1 proteases are among the protein antigens that are immunoprotective against Streptococcus pneumoniae infections. Selective mutations in the functional sites of these proteins may be used to improve the protective efficacies of these proteins as vaccine candidates. Genomic islands encoding virulence factors of pathogenic bacteria have been designated pathogenicity islands. Pathogenicity islands were first discovered in uropathogenic Escherichia coli. Advances in molecular biology, cell biology, and several other areas of science have changed the way we understand the mechanisms in which microbial pathogens interact with their hosts. This trend is set to continue with the advent of microbial genome sequencing, in vivo gene expression analysis, and other related techniques. However, despite these powerful approaches, the basic concept advanced many years ago by Smith that pathogenicity of virulence is a multifactorial property that consists of 5 basic steps is still valid today. The molecular basis of virulence can still be considered under these 5 headings. First, attachment to the host via mucous membrane. A good example of this fact is the interaction of respiratory pathogen Streptococcus pneumoniae, the pneumococcus, with the host cells. At least 4 types of receptor have been proposed for the pneumococcus, and the interaction with these receptors depends on the phenotype of the organism. Second, entry into the host. Usually, entry of Shigella into cells is triggered by 3 gene products: IPA-B, invasion plasmid antigen B, IPA-C, and IPA-D. IPA-B is a hemolysin responsible for the release of Shigella from the vacuole into the cytoplasm, where the organism then moved by the polymerization of host actin under the influence of the intracellular spread gene ICSA. This motility results in the generation of protrusions from one cell into the next. The protrusion is clipped off to form a vacuole within the next cell. The double membranes of this vacuole are then lysed by the product of the ICSB gene. The organisms are released into the cytoplasm, and the cycle begins again. Number 3. Multiplication within the host. Rapid multiplication leads to acute disease, whereas slow multiplication may be advantageous in chronic disease. The environmental factors and nutrients that determine growth rate in tissues remain largely unknown. Most detailed studies on factors controlling growth in vivo have concerned molecular mechanisms for overcoming iron restriction. Pathogens attempt to obtain iron different by one of several mechanisms, including A. From hemoglobin as hemin or hem, for example, as pneumonia. B. Directly from ferrated transferrin or lactoferrin, for example, Neisseria gonorrhea. C. Indirectly from iron-binding proteins by the production of siderophores, for example, in E. coli 12. D. From intracellular iron stores, for example, Mycobacterium tuberculosis. Number 4. Interference with host defense systems. To survive within the host, the pathogen must either prevent the immune response or circumvent its action. There are many virulence factors associated with interference of host defense, including polysaccharide capsules, protein toxins, and lipopolysaccharides. Polysaccharide capsules interfere with the processes of phagocytosis and complement mediated bacterial killing. The importance of the capsule in the virulence of some organisms has been demonstrated at the molecular level. A non-capsular mutant of the pneumococcus generated by transposon mutagenesis was 1 million times less virulent than its capsular parent. The molecular mechanisms of activity of capsular polysaccharide still remain to be defined. It is known that C-allele groups on capsular polysaccharides of E. coli K1 and group B streptococci may prevent the activation of the complement pathway by these organisms. Antibodies to capsular polysaccharide usually confer protective immunity through opsonization of bacteria promoting phagocytosis. Encapsulated pathogens are still major causes of human disease due to the production of diverse capsular types, for example, pneumococcus, or to the capsule being non-immunogenic, for example, E. coli K1 and Neisseria meningitidis group B. The non-immunogenicity of these organisms may be due to the molecular structure of the polysaccharide subunit being similar to sugars found on host cells. Number 5. Damage to the host. There is a wealth of information in the literature concerning the molecular action of bacterial protein toxins having broad spectrum of action. ATP ribosylation, protease activity, induction of inappropriate cytokine production, or activation of the complement cascade, stimulation of immunopathological reactions, which may also lead to damage to the host. These 5 stages are not mutually exclusive. The factors produced by pathogens that mediate these steps are turned to determinants of microbial pathogenicity. An understanding of the events that occur at the molecular level, both at the action of individual virulence factors and in the coordinated regulation of virulence as a whole, is a continuing aim that will be aided by the new trend of total genome sequencing of pathogens. Comprehension of the molecular events involved in the disease process may allow us to generate new weapons to use in the continuing battle against infectious diseases. Please feel free to leave your questions and comments in the forum, and we'll